She graduated with a Bachelor of Science in Animal Biology and a minor in Biotechnology last year from the University of Guelph, and has had successful summer internships with the Ontario Ministry of Agriculture, Food and Rural Affairs, and URIS. She's currently working on investigating the genetic relationships between colostrum traits, feed efficiency, and growth traits in Canadian Holstein calves, and will later work on estimating the impact of the transition period on feed efficiency in Holstein cattle. So thanks for coming in with us today. And I'll give you this, which is meant to be like around your neck if you want that. Yeah, but just be careful, you don't want to leave.
Just don't put on the floor.
Yes.
Okay. Is it best to sit or stand?
Stand.
Okay. Awesome. Okay. Thank you so much for the introduction. So the paper that I will be discussing today is called The Genetic Parameters of Colostrum and Calf Serum Antibodies in Swedish Dairy Cattle. So to start with some background, dairy calves are born immunologically naive due to the structure of the dairy cow's placenta. So ingesting colostrum after birth provides an immediate source of maternal antibodies for dairy calves and helps them acquire passive immunity and assist with their digestive tract development. Passive transfer, which is the process of how they're absorbing these antibodies, is the absorption of maternal immunoglobulins present in the colostrum they digest through the calf's small intestine 24 hours after birth. So colostrum quality is typically represented as the concentration of antibodies, so immunoglobulin in this case, and in particular IgG, or it can be represented through serum total protein, or STP, which is a measure of the total protein found in the blood of a calf and is also used as an indicator for successful passive transfer. If the amount of transferred antibodies is not sufficient 24 hours after birth, then failure of passive transfer, or FPT, can occur. And this has about a 14 to 60% prevalence on Swedish dairy herds. So this little diagram shows that when calves have colostrum with less than 10 grams per liters of colostrum with IgG, or if their calf serum blood concentration is less than 5.5 grams of deciliter in their serum, then they're at a twofold higher risk of morbidity and death. So therefore, colostrum quality plays a major role in preventing FPT in dairy calves. These are some of the tools that are used to assess colostrum quality that were discussed in the paper. So the three that were mentioned were radial immunodiffusion assay, or RID, enzyme-linked immunosorbent assay, or ELISA, and a refractometer. So the first two are direct measures of IgG, and a refractometer is an indirect measurement based on the total protein content in the colostrum sample. There are a couple pros and cons for each tool, but what I want to highlight is that a refractometer is cost-effective, easy to use, and it also has a moderate correlation with RID, and this is a common tool used on farm to assess colostrum quality. When talking about colostrum quality, it does vary largely between cows, even between animals that are from the same farm and the same breed. And some of the literature is suggesting that genetics may be potentially causing some of this variation. And despite its significant role in calf health, few studies have investigated the genetics associated with calf antibody uptake from colostrum. Furthermore, previous literature also suggests there's a genetic component in the antibody content itself in the colostrum. So two studies I want to highlight. There was one done in 2019 where they estimated a heritability of 0.27 per Brix percentage in Holstein colostrum, and another study done in 2021 where the heritability was 0.36 for calf serum IgG content at 24 to 48 hours after birth in Charolais calves. So therefore, understanding the genetic relationship between antibodies in colostrum and calf serum will aid in reducing FPT and also improve calf health. So there were three objectives for this study. The first one was to estimate the heritability of calf antibody uptake for colostrum using calf serum antibody traits and STP as an indicator trait. The second was to estimate heritability of antibody traits from colostrum, milk, and serum using Brix percentage as an indicator. And the final objective was to determine how these traits correlate with each other and also with milk production traits later on in life. In terms of the methods, so samples were collected on 3 experimental farms in Sweden with a total of 1,340 cows being included in the study. And you can see the breakdown of the breeds. So there was Swedish Reds, Swedish Holsteins, and then a crossbred breed between those two, with parities ranging from 1 to 6 for those animals. During the sampling period, 504 of these animals calved twice and 29 calved 3 times. Sampling of first milk colostrum was carried out by farm staff with 1,711 records collected. 886 calves, which had been calved by cows with a matching colostrum sample in the study, were sampled. 59 of those calves could not be linked to a pedigree record, so they were removed. And calves were sampled between ages 1 to 12 days, and they had their birth weight, volume of their first meal, the time of their first meal after birth, and also their colostrum donor recorded for them. And all calves were given milk and colostrum as their first meal. And you can see the breakdown of the number of records based on the variables being collected and the periods that they were collected in this table. So we have the Brix percentage, the total IgG per liter, and then NAB, which stands for natural antibodies. And I'm going to touch on that in the next slide where I explain a little bit more about them. And then STP, and also serum IgG content in the calf serum category. For phenotypes, a Brix refractometer was used to analyze colostrum samples. Again, this is approximate percentage, and the scale they used was 0 to 53% of the total solids as an estimate for the IgG content. An optical refractometer was used to estimate to analyze calf serum samples with total solids to estimate STP. And then an ELISA test was used to measure the total IgG levels in the colostrum and calf serum. In terms of the antibodies, which are standing for NAB, a 2-step ELISA test was used to measure the antibody isotypes of interest, which were IgM, IgG, and IgA. And then 2 antigens were used to come up with 6 genetic traits that were analyzed in this study. So the 2 antigens were KLH and MDP, which 2 comes with 6 traits that were being analyzed in this study. In terms of statistical analysis, it's broken down based off the cows and the calves. So first, the variance components for the genetic effects, permanent environmental effects for the colostrum traits, and maternal effects for calf serum were estimated with an animal model using AS-REML. The pedigree data contained around 29,000 records with 20 generations, and the models are organized based off the cows and calves, like I mentioned before. The formatting seems a little off here, but I want to highlight the covariates that are included in each of these models. So the C2CS covariate in the colostrum traits repeatability model represents the effect of colostrum sampling time after calving in hours. C2S in the second model is the covariate describing the effect of sampling time the days before or after calving. And the AFC variable in this last model for cow production traits represents the effect of age at first calving in months. So these are the 3 models that were used to estimate the observations of the traits of interest for cow colostrum, cow production, and also cow serum. In terms of calves, the covariates that I want to highlight include COL, which is a covariate describing the absolute amount of antibodies received from the matching colostrum trait. There was also BLD, which is a covariate describing the time from calving to blood sampling. And finally, BWT, which is a covariate of the calf birth weight in kilograms. And finally, heritability, repeatability, and maternal effects were all estimated for the previous traits I mentioned. Going into some results. So for the tables that I'll be presenting for the next few slides, the numbers that are bold are significant, and then the numbers in brackets represent the standard errors that were calculated. So this first table is looking at the heritability, maternal effects, and phenotypic variances for the calf serum traits. And you can see that the traits showed low to moderate heritability ranging from 0.25 to 0.59. What I really want to highlight here is that STP as an indicator trait was not significant, and it also has a really low heritability. You can see that the maternal contributions were moderate for each of the respective traits, except for KLH IgA, so one of those antibody traits that were being looked at. And the maternal effect for this study was the non-genetic contribution of the dam across the different calvings for all of the calf serum traits. And it means that the maternal contribution to variation in these traits was around 17 to 37%. So I touched on this a little bit in the discussion, but for calf serum antibodies, the greatest non-genetic maternal contribution is the content of the colostrum, which was already accounted for in the model. And what they hypothesized was other colostrum components may be influencing how well the calves are actually uptaking and absorbing the antibodies, such as fat, protein, peptides, or growth factors. And it seems plausible that the component content of those different varies from different cows, and that might directly or indirectly affect antibody uptake in the calves. This second table is looking at the heritability, repeatability, and the phenotypic variances for the cow sample traits. And what I want to highlight is that the heritability for Brix as an indicator trait and also for total IgG were significant, moderate, 0.31 and 0.2 respectively, and also they had moderate repeatability. And then these third and the fourth table are looking at the genetic and phenotypic correlations between Brix and colostrum traits. And you can see that Brix had a positive genetic correlation ranging from 0.49 to 0.73 with the respective traits of interest. And all the phenotypic correlations were also positive and ranging from 0.48 to 0.70. Brix was positively correlated with all of the traits, including total IgG content, which means that it could be a promising indicator trait for selection of higher quality colostrum. Table 5 and Table 6 are looking at the phenotypic and genetic correlations between STP and calf serum traits, so STP being the indicator trait of interest here. And the phenotypic correlations range from 0.37 to 0.67 with the respective traits. Genetic correlations range from about 0.47 to 0.91. Unlike Brix percentage for colostrum traits, STP as an indicator trait did not show significant genetic correlations with any of the calf antibody traits. And there was also no significant genetic correlation between STP and Brix percentage. This final table is looking at the genetic correlations of the two indicator traits respectively, so Brix for colostrum and STP for calf serum with the milk production traits. And you can see that there was no significant genetic correlation between the two indicator traits and any of the milk production traits. There's also an unclear relationship between calf serum IgG and milk production performance later on in life. So in conclusion, the calf serum traits are heritable, but STP showed a low heritability and no significant genetic correlations with the traits of interest in this study. There was a significant maternal contribution to calf serum antibody content. Brix percentage has positive genetic correlations with the investigated antibody traits, which means it could be used as an indicator trait to select for higher quality colostrum. And further studies are needed to investigate whether milk production traits are genetically correlated with Brix. And that's everything I have for the paper. If you have any questions, please let me know.
Can you hear me?
Yes.
Okay, perfect. I do have a question. So I was reading over your paper earlier, and in their conclusion, they said that it would be a good way, or it'd be something good to include in, breeding programs. So selecting for this in breeding programs. What's kind of your take on it? Do you think it would be a good thing to kind of spend time and money on to add to a breeding program when at least a lot of the farms that I know of use like replacement, so like powdered colostrum?
For sure. I think with the powdered colostrum, I've seen it be used as both like we're using it independent of the colostrum that's being produced as a form of just giving that to the calf. And then also I've seen it be used as a supplement to maybe lower quality colostrum. So I think the benefit of including this in a breeding program is perhaps the economic cost associated with buying that powdered colostrum, and then also with the economics of having staff go around and feed those calves and make sure that the colostrum quality is adequate versus knowing that through selection with a breeding program, they are producing higher quality colostrum. I think that second option where we're including this through the genetics pathway versus nutrition and supplementing it, I think it's a much more beneficial method of improving colostrum quality.
Thank you.
Great, thank you. Do we have any other questions in person, online? If you're online and would prefer, you can type it into the chat as well. Colin?
So when you're second for these, is that on? Is that green? Yeah.
Yes, we agree.
Yeah. So the idea of selecting on these traits is, I'm guessing the overall objective is to reduce disease incidence.
Yeah.
So can you describe to me what the advantage that these traits have versus just selecting on the disease traits themselves? So for example, my work.
Yeah, I think that is a good question. At least from this paper's perspective, it seems that the failure of passive transfer is a bigger issue in Swedish dairy farms. I'm not sure how big it is like in Ontario or across Canada, but I think selecting for higher quality colostrum using these traits, especially traits that are easy to collect phenotypes from. So for the Brix percentage, for example, I think that makes more sense in managing it through that method, and then maybe also further down the road selecting for those calf disease traits. But I think the benefit of having a trait that's so easy to phenotype and collect, especially from a farmer's perspective, like they don't need to bring somebody else in to collect that data. It doesn't really cost them anything extra. I think that's where the benefit of this paper really comes through.
Perfect. Thank you.
I was wondering about the Brix.
Yeah.
And what's kind of like, how do they take those measurements, and why would you would you use the bricks rather than the other methods that you mentioned?
For sure, yeah. So there are these are the three like most common that I've seen, at least from the dairy industry. I'm not sure if there's any other that are being used, but the benefit of the bricks refractometer tool is it's portable, it's easy to, I don't think it's very expensive, and also doesn't require a veterinarian or a laboratory setting compared to like the ELISA and the RID assay. So how the bricks test works is you like pipette a sample of the colostrum on like this little plate that's weighted, and it measures it, and then it does the indirect measurement of the total solid content, and it calculates it based on like a percentage scale. So for example, for this one, it was 0 to 53%. Typically, what I see farmers say is anything that's like 22% or higher is considered good quality colostrum, and they would just feed that to the calf. Anything lower than that, they're either not feeding it or they're doing what Kelly mentioned, which is supplementing with a powdered colostrum supplement so that they kind of still meet that requirement without having to like toss a bunch of colostrum out. So it's a very handy tool to have on farm. A lot of farmers are already using it to test, but they're not really doing anything with that information. They're just being aware of the fact that the bricks percentage is this much.
Right. So the data it's collected.
Yes.
And what else was I going to ask?
Can I ask one question from Avalon?
Yeah, go ahead.
Avalon, I was wondering whether there is a correlation between this IgA concentration and the total solid content in the colostrum. If you mentioned this in your presentation, sorry if I missed that.
No, you're so good. I am not sure about the genetic correlations. I know that the one correlation that they wanted to call out was there's no significant correlation between STP, the serum total protein, and the IgG content, and then also STP and the bricks percentage measurement. But I know that when in the paper they talked about the benefit of bricks is that it does have a high correlation with the RID measurements, which is based off of total IgG content. So there might be something there in further research, but kind of in this paper, I don't know if they actually explored that, or if they did, I don't recall if it was significant or not.
I also have a really quick question, just because
sure.
I'm not super familiar with the topic, but is the bricks the only way that they can collect measurements for colostrum, or is there like any other way?
Yeah, so for colostrum, the bricks test is the most commonly used, I think, and also on farm, it's the most immediately applicable. There are those two other assays that I mentioned, but you can also look at calf serum as a measurement of like antibody uptake and passive transfer in calves, and that's where that STP trait comes in. The other problem with the serum total protein trait is it again requires a veterinarian or somebody who's taking all those blood samples and then testing them in a lab. So the bricks percentage really does kind of come out on top in terms of like easy to measure, not very expensive, doesn't require a lot of labor. But you can use the bricks percentage measurement or the other two assays to do colostrum. And then if you want to perhaps look at antibody transfer in another way, you could look at the calf serum traits.
Okay, perfect. Thank you.
Yeah, no problem.
All right. Any last questions for Avalon online? All right. I think you're good.
No problem.
Great. All right, so now we'll move on to Emily. Emily, I'll let you share your screen so we get that up and running first.
For sure. Okay. Can you see it?
Yes, just a minute here. All right. Okay, I think we're good. So now we have Emily, who is presenting the paper titled Residual Feed Intake, a Limiting Economic Factor for Selection in Poultry Breeding Programs. So Emily Lomas graduated with her undergraduate degree in biology from Wilfrid Laurier University in June 2023. And she is currently pursuing her Master's of Science in Animal Breeding and Genetics where her project focuses on investigating the genetic correlation between postmortem muscle activity and meat quality traits in turkeys. Her ultimate goal is to gain admissions into the Ontario Veterinary College at the end of her master's degree in 2024. So thank you, Emily. And I'll give the floor over to you.
Thank you so much. So good afternoon, everyone. As Kristen mentioned, I'm Emily, and today I'm going to be providing a paper review of residual feed intake, a limiting economic factor for selection in poultry programs. So it's important to mention that this paper itself is actually a literature review rather than a primary research study. So I'll be describing the findings that were comprised in this paper by the authors. So I wanted to begin by providing a brief overview of the flow of my presentation. I'll begin by providing an introduction of the paper and its main points, followed by a description of both the objective and hypothesis of the paper. Next, I will introduce the material and methods section, followed by the results and discussion. Finally, I will finish by presenting the primary conclusions of the paper. If there are any questions along the way, feel free to just interrupt me as well, or just ask at the end of the presentation. So first, I'll introduce what residual feed intake is to create a good foundation moving forward for the presentation. As defined in the paper, residual feed intake is an estimate of feed efficiency independent of the level of production, and it is the difference between the observed feed intake and predicted feed intake in an individual. So why should we measure residual feed intake? Feed tends to be one of the main variable components that affect the productive process of animals in all farming, and in this case, poultry farming. It constitutes a substantial portion of the overall expenses in the industry and provides essential nutrients for growth, maintenance, and reproduction. Variations in feed quality, quantity, or composition can have profound effects on the productivity of animals. Animals that efficiently convert feed into desired products exhibit better overall productivity, while inefficiencies can lead to increased costs and reduced profitability. Given that feed expenses represent a significant portion of total costs, improvement in feed efficiency can have substantial economic benefits, helping to reduce production costs and enhance profitability. So in this paper, there was a variety of data that was comprised from various other studies. In each of these studies, the primary species that were investigated were chickens, quails, ducks, and turkeys. When a bird has a negative residual feed intake, or R negative, it means that it consumes less feed than expected for its maintenance and productive needs. In other words, it achieves its body weight and production performance while consuming less feed, making it more efficient in converting feed into growth or egg production. This efficiency will lead to lower feed costs and impact overall profitability, as discussed in the previous slide. Now, depending on the study that was conducted, either egg producing or meat producing lines were examined. There are two equations that you can see at the bottom of the slide. The top one denotes the equation used for egg producing lines and the bottom for meat producing lines. For the egg producing lines, the EFI is the expected feed intake of hen I, where BWI to the power of 0.75 is the mean metabolic weight of hen I, EMI is the egg mass production of the hen, delta BWI is the body weight gain of the hen, A, B, and C are partial regression coefficients, and D is the intercept. For the meat producing lines, it's very similar, where the EFI is the expected feed intake of the broiler I, where MBWI to the power of 0.75 is the mean metabolic body weight, BWGI is the body weight gain of the broiler, B1 and B2 are partial regression coefficients, and A is the intercept. Now, the primary objective of this paper was to explain the concept of residual feed intake and underscore its importance in gauging feed efficiency within poultry production systems. Unlike conventional measures like the feed conversion ratio, which only assesses efficiency at a specific production level, residual feed intake accounts for maintenance requirements and thus provides a more comprehensive understanding of feed efficiency. Furthermore, the paper aimed to highlight the potential of genetic selection in enhancing feed efficiency through the manipulation of residual feed intake. By selectively breeding animals with lower RFI values, poultry producers can cultivate a more efficient flock that requires less feed to achieve the desired level of production. This leads me to the overarching hypothesis of each paper. Incorporating RFI into poultry breeding programs can lead to substantial enhancements in feed efficiency, productivity, and environmental sustainability within the poultry industry. So I want to break this hypothesis down a little bit. It suggests a couple of different things. So firstly, the hypothesis implies that integrating RFI as a selection criterion will lead to improvements in feed efficiency, as birds with lower RFI require less feed to achieve comparable levels of production output. Secondly, it would suggest that selecting for lower RFI will enhance overall productivity by optimizing resource utilization and minimizing feed wastage. Finally, it proposes that breeding for lower RFI will contribute to environmental sustainability by reducing greenhouse gas emissions and pollutions associated with poultry production. So overall, the hypothesis posits that incorporating RFI into breeding programs will yield multifaceted benefits for the poultry industry. Now I'll present the materials and methods collected in the studies and presented in the paper I had chosen. So there were various breeds of chickens, turkeys, ducks, and quails, as I had mentioned. Of the ones that were reported in this paper, there were 8 chicken breeds, no specific turkey breed as it was just labeled turkey, 2 duck breeds, and 1 breed of quail that was separated into gray and white birds. So of these species and breeds, they were further separated into either meat-producing or egg-producing lines. The specifics of which lines were which were not actually noted in the paper, further than mentioning that the separation of egg and meat did occur. There were several types of measurements that were also taken from the birds, such as feed intake, body weight, egg production, if applicable, blood samples, and post-mortem examination of corpses where the carcass, liver, heart, gizzard, leg weight, and muscle magret weight were measured. Also in this paper, there were 4 main tables that were reporting important information. So the first was the RFIs of different poultry populations. This table presented the computed RFI values for different populations of poultry, categorized based on production type, line or breed, and species. It provides equations that predict the expected feed intake based on metabolic body weight gain, and egg mass production over specific periods. These equations offered insights into the factors influencing feed efficiency across different poultry varieties and can aid in the selection of birds with lower RFI values for improved efficiency. The second table was the genotypic and phenotypic correlations between RFI and productive traits. This table highlighted the relationships between RFI and traits such as body weight, feed intake, feed conversion ratio, egg production percentage, and egg weight. These correlations provided valuable information for understanding how RFI influences overall productivity and can also guide breeding programs aimed at improving feed efficiency and production traits simultaneously. The third table, which I reproduced from the paper on the right-hand side of the slide here, this one illustrates the phenotypic correlations between RFI and blood parameters in quail populations. So it includes correlations with parameters such as total protein, albumin, globulin, cholesterol, triglyceride, hemoglobin, red blood cells, hematocrit, and thrombocytes, which are platelets. These correlations offer insights into the physiological aspects associated with RFI and its potential impacts on the health and metabolic status of quail. You can notice that for the white strain, the RFI was negatively correlated with all hematological aspects, while in the gray strain, the RFI was positively correlated with red blood cells and hematocrit. I've highlighted all of the elements of the hematology in green in that table just to make that easier to visualize. So now there are a couple things that this could indicate. First, that this could suggest that birds with improved feed efficiency in the white strain may have better overall health and metabolic status, which is reflected in their hematological profiles. It may also suggest that birds with higher RFI in the gray strain tended to have higher levels of these hematological parameters, which may indicate a potential adaptation to physiological stressors or metabolic demands in birds with higher RFI within the gray strain. Finally, it could also suggest variations on the regulatory mechanisms governing all of the hematological parameters between the white and gray strains in response to differences in feed efficiency. Finally, in the fourth reported table, the phenotypic correlations between RFI and carcass traits in quail and duck populations was presented. So that one included correlations with traits such as carcass, liver, heart, gizzard, leg, and muscle magret weights. These correlations shed light on the relationship between RFI and carcass composition, indicating potential implications for meat yield and quality in duck and quail population systems. In this slide, I compiled a concise summary of the key findings regarding heritability, genotypic correlations of RFI, environmental conditions, and immune response. So firstly, it highlights that RFI exhibits moderate heritability across various poultry populations, with estimates ranging from 0.21 to 0.49. Unfortunately, this paper did not indicate which estimates were from which species or breed, so all we can deduce is that there is low to moderate heritability across the 4 species of birds reported. This heritability would suggest that genetic factors do play a significant role in determining feed efficiency. Secondly, the genetic or genotypic correlations of RFI reveal interesting relationships with other traits. For instance, RFI showed negative correlations with hematology in white quails, while it is positively correlated with certain hematological parameters in gray quails. You can also see it shows a negative correlation between RFI and body weight gain, carcass percentage, and breast muscle percentage across all birds reported, while it shows a positive correlation to egg weight, number, and mass, as well as feed consumption and body weight across all birds. The table also emphasizes the environmental implications of improving RFI. So it suggests that enhancing feed efficiency not only benefits productivity, but also contributes to environmental sustainability by reducing emissions of ammonia, carbon dioxide, and methane. Lastly, insights to the immune response were provided as well, indicating that there were no significant differences in antibody formation between birds with different RFIs, so either positive or negative. However, non-efficient birds demonstrated higher immune system responses under unchallenged conditions, suggesting potential trade-offs between feed efficiency and immune function. Overall, this table offered valuable insights into the multifaceted nature of RFI and its implications for poultry breeding and management. So in conclusion, the paper provided valuable insights into the concept of residual feed intake and its intricate relationship with various productive traits in poultry. It emphasizes the importance of incorporating RFI as a negative trait in poultry breeding programs. By selecting for low RFI, breeders can aim to produce more efficient poultry breeds characterized by reduced feed costs and increased productivity. Additionally, the adoption of RFI as a selection criterion holds promise for enhancing environmental sustainability in poultry production. This is because poultry with low RFI tend to emit fewer pollutants such as ammonia and greenhouse gases, thereby mitigating the environmental impact of poultry farming. Overall, the findings underscore the significance of RFI as a key indicator of feed efficiency and highlight its potential to drive improvements in both economic and environmental aspects of poultry production. And that is all.
Thanks, Emily. Do we have any questions for Emily? Go ahead, Avalon.
I just had a question about the negative correlation with RFI and the breast muscle percentage. How does that impact like the economics of the actual carcass? Like I'm not very familiar with birds, so I am sorry, but is that a bad thing if it's impacting like carcass development and how that bird is being priced, or is there any concern with that?
Yeah, so in the paper specifically, they didn't mention about like the economic impact of that. Are you just asking like I'm trying to reword this, like how is the RFI correlated negatively with breast muscle?
I was just wondering if there's like a drawback. So if we're selecting for more feed efficient birds, is there a concern that we're then maybe inherently breeding birds with smaller muscles in their, like with smaller breast muscles? And does that impact like how the carcass is priced, or is it different for poultry?
Okay, yeah, so definitely it would be the same with poultry with, you know, bigger yield of breast meat would be more beneficial cost-wise. I think that there is a little bit of a trade-off between the economic benefits and the feed efficiency. So it would be kind of determining if you're wanting to lose more money in less breast meat or in paying more for your feed. So I think it just kind of ends up being determined by each farmer to see if one kind of offsets the other in terms of how much they would be losing per se. But yeah.
Awesome, thank you.
I have a question, Emily.
Yeah.
So based on the positive and the negatively correlated traits and all that, if in your opinion, do you think it would be more beneficial to kind of implement this for meat producing birds or more so for egg laying birds?
So based on the paper and what I was reading, it seems to be more beneficial for egg producing birds because there was that negative correlation between some of the meat yield and the feed efficiency. So I think where I mentioned to Avalon with the kind of economic offsets between the feed efficiency and the yield of meat, I think, yeah, it would end up being more beneficial to do it in egg producing lines over the meat producing lines.
Okay. And do you think if you were to implement it on the meat producing lines, do you think it would like I couldn't hear what you were telling Avalon too much. It was cutting out a lot. But do you think there would be more of a benefit even in the meat laying or the meat lines to offset the cost of the food, even though you're losing money in the meat?
I personally would be kind of curious to see if we could implement both like breeding for feed efficiency as well as meat yield. Like, I don't know if this has been done or not. I didn't really look too much into it, but I think that would probably be the best option, would be to try and increase meat yield while increasing feed efficiency, just for the best economic benefit.
Emily,
Yeah.
I have a question about the RFI. I don't know. I don't remember if you showed it in the presentation, how the phenotype was actually used, because I was just thinking about Avalon's question for the negative correlation with breast muscle, and it doesn't make much sense to me. So I just wanted to understand if they didn't invert RFI so that animals with a smaller intake are more efficient and then are converting into more muscle.
Yeah, that would make more sense. They didn't have, so the paper itself was not super descriptive. They reported a lot of genotypic correlations instead of the phenotypic. So I might personally look into that a little bit afterwards and see if I can find anything about it, because what you're saying totally makes sense and I agree. So I'll look into it and see if I can find something.
Okay.
All right, do we have any other questions online, in person? Doesn't look like it. I was wondering, so kind of the way you were describing it, they haven't been selecting for feed efficiency in poultry at all? Poultry breeding programs?
So it doesn't seem that this has been implemented as like a primary concern. I think right now the primary thing that's being bred for is meat yield or like egg production rather than feed efficiency itself. So I think this paper was, mostly kind of pushing to say that we could do all of the above and work to try and get feed efficient animals while also producing higher meat yield and egg production as well.
Okay. And did it describe how you collect feed efficiency in poultry? I just know how it's done in like cattle, but not in birds.
So again, what this paper mentioned is that they were calculating it based on these equations here. So it depends on the egg production and the meat producing lines. So they would be incorporating different measurements that they had taken from the birds individually, and then calculating individual RFI that way.
Okay.
Yeah.
All right. Last call for questions. No more. All right. Thank you, Emily. All right, and thank you everyone for joining us today. And thank you, Avalon, again, and Emily for the very nice presentations. And we'll be back not next week, but the following Friday.
Thank you. Thank you very much.
Thank you, Kristen.